Thanks a lot. I'm going to apologize in advance for sort of sounding hoarse, and hopefully my voice will laugh and I won't start coughing too much, but I've gotten the bug. And I would like to say that it's really great to be here, and I'm really excited with the people I've talked with so far, and I'm looking forward to talking with more of you. And I want to share a couple of talks. It's really fun to be able to give two talks, not one. And I'm going to sort of grind my axe and sort of think about conservation behavior. So challenge me. Let's talk about conservation behavior. A friend and I, Esteban Fernandez-Yersic, are writing a textbook right now called A Primer on Conservation Behavior that Cinauer is going to publish if Esteban ever writes his 6 chapters. But we're enthused about this. And really what I want to do is, with this talk at least, is talk about some lessons I've learned from marmots and potential take-home lessons that might extend beyond marmots to other systems that people might be working with. And with tomorrow's talk broadly about anti-predator behavior and trying to increase reintroduction success by having knowledge of behavior, specifically anti-predator behavior. So before I get started, when I say conservation behavior, what I'm talking about is the application of behavioral knowledge to solve wildlife conservation problems. And often, this knowledge can help increase the success of captive breeding programs for conservation. Sometimes it can increase the success of reintroductions or translocations to recover populations, because we know most of these fail. Some people argue we shouldn't even do them because of the ethical costs associated with failure, which means animals die. And in some cases, we might be able to manage natural behavior to manage natural populations. So it's really a mix of captive stuff, wild stuff, and at some level, really the sky's the limit. And I think we're still in the sort of the development of this field, and I think that the goal is to make a toolbox. But importantly, rather than saying this is the bee's knees, which I don't think it is, I'm going to put all of this in a perspective of adaptive management. As you all know probably, there's active adaptive management where we're creating management plans that are modified based on the results of well-designed experiments that collect data on factors or variables that are demonstrably important for conservation or management problems. And I believe that if possible, it's good to design active adaptive management programs when to study some of these things that I might be talking about and to study the efficacy of a conservation behavior suggestion or mitigation. And then there's also passive adaptive management, where managers use historical data or data from uncontrolled experiments to sort of come up with best guess management recommendations, the fate of which may be studied in sort of a less controlled fashion. I think that, if possible, what we want to really be doing is active adaptive management when we're sort of testing the efficacy of conservation behavior suggestions. And with that, let's get on to the subject of the main subject of the talk, which are marmots. So marmots are large ground-dwelling siluriid rodents, and for the past 20 years or so, I've studied 8 of the 14 species. They have a whole Arctic distribution. They're found around the northern hemisphere. Most of them live in nice alpine environments. Some of them live in what I call Central America, the Midwest. Others live in the sort of Central Asia, or the steppes, which are sort of an equally flat, topographically challenged environment. But many of them live in really nice aesthetic environments. And while these environments are either similar or different, the marmots themselves illustrate a remarkable amount of social variety. And one of the things I've been studying is the evolution of sociality and the evolution of communication. Now, what's interesting about marmots is that many of them have issues. So the Vancouver Island marmot, there are fewer than 100 in the wild, I think, right now. There's an active captive breeding program and reintroductions. Many of these reintroductions have been failing because of predation. The captive breeding program was almost a non-starter because of animal husbandry and management issues. Behavior has been important in some of these managing this population. The Men's-Beers marmots, which is a Central Asian species that's listed as vulnerable from habitat loss and hunting. The long-tailed marmot in Afghanistan, Pakistan, Tajikistan. According to the IUCN, it's near threatened from habitat loss and hunting. There were a lot of them where I studied them in Pakistan. The steppe marmot, which has saved Russians and Soviets for years during periodic famines. People eat them. Also, a lot of information is known about them because they're plague vectors, and there's The KGB was very interested in marmots. There's a lot of population biology about marmots and plague and disease ecology and KGB colonels that used to study marmots for bioweapons research. But this species is actually subject to substantial hunting now, and I mean, it's sort of like the decline of prairie dogs, really. And then not listed, but still of concern, is a Mongolian species, which everyone I talk to who works in Mongolia is terrified about the decline in the population of some of the Mongolian species of marmots. The black-capped marmot, which has a disjoint distribution with very small population sizes. One population lives near Lake Baikal, the other lives on the Kamchatka Peninsula. And the Olympic marmot, our own marmot, endemic to the peaks of the Olympic Peninsula. This is a place where global warming will ultimately eliminate their habitat meadows. But more importantly, in the last 20 or so years, their populations have declined too, immediately south of the mountaintops on which the Vancouver Island marmot lived. And don't know why. There's a colleague of mine who's studying them. Climate change, changes in distribution of predators, a little bit of both, who knows? So a lot of these marmots have issues, and captive breeding and reintroduction are going to be important things to save marmots. I mean, can you imagine a life without marmots? It would be going to alpine meadows and not being whistled or chirped at. Very, very sad. But importantly, marmots are also important prey to top carnivores. So if you value carnivores and carnivore conservation, you should value marmots in places where they live. Where they live, marmots may be the most important summer food source for snow leopards. Wolves eat them during the summer. Foxes eat them. We have one fox who's eaten most of our marmots in the past couple of years. Eagles, bearded vultures, brown bears, badgers, mountain lions, lynx. You know, all of these animals like to either feed their kids a nice, fat, juicy marmot or try to fatten up for their own winter survival by eating a nice, fat, juicy marmot. So there have, as I said, I alluded to before, there has been active conservation management of several species, and some of these have been successful. So the steppe marmot, there's been a lot of experience breeding them in captivity, both for conservation reasons and for fur. I went to a fur farm outside Moscow a number of years ago, and the only happy animals in this fur farm were the marmots. Steppe marmots are highly social, and they were just cheek to jowl and looked like a high-rise, you know, rabbit hutch really, but a marmot hutch. And you know, they were all really healthy looking with great fur and more or less no stereotypies and seemed to be okay. All these other animals, the foxes, the stoats, the mink, you know, horrible stereotypies. Really, fur farms are scary places. But in any event, there also have been translocations where techniques have been developed to catch whole colonies of marmots and move them around. And the Russians and people in the former Soviet Union have been interested in moving them around. Alpine marmots in Europe, the Europeans really love their marmots, and alpine marmot went extinct in the Pyrenees, and there was a successful translocation to repatriate marmots to the Pyrenees. And as I said, there's an ongoing program to save the Vancouver Island marmot, which seems to be working, albeit slowly. Now, captive breeding programs, so the main point is that to design proper reintroductions or captive breeding, future management will be more successful, I think, if we have a good understanding of natural behavior in the wild, and that captive breeding programs also will be enhanced by a fundamental understanding of behavior in the wild. So what I'm going to really talk about today are lessons I think we may have learned, or lessons I suggest we've learned with respect to anti-predator behavior, habitat acoustics, stress, predation risk, individuality, and complex sociality and reproductive suppression. So it's really a wide variety of topics that I think have conservation importance when we're thinking about captive breeding and reintroduction of marmots. Now, while I've worked all over the place studying marmots, many of the data and many of the studies I'm talking about today come from a long-term data set that Ken Armitage started collecting in 1962 at the Rocky Mountain Biological Laboratory. These marmots are one of the longest-running continuous studies of the population biology of a free-living mammal pretty much anywhere. So from these studies, we know, and from a lot of Ken's work, we know about some of the factors that influence population demographic trends. And we know that marmots are seemingly emerging earlier since it's been getting warmer. I did some work in the 90s, and then I began in earnest taking over the study in 2001. And we've both been continuing population biology questions and expanding it into the study to also look at a variety of behavioral questions. I think it's a really productive system, and I think the insights you get from long-term studies are particularly important and precious. So the first lesson I want to talk about is that anti-predator behavior evolved as a game between predators and prey. This is a fox, this is a marmot. This fox would like to eat this marmot doesn't want to get eaten by this fox. Long history of evolutionary games between predators and prey, and what are some lessons from this? Well, if you think about alarm calls animals produce when they get scared by predators, these could be directed to the predator, maybe to signal detection and thus to discourage pursuit. So a scared animal could be saying, "I've seen you," and if by saying "I've seen you," predators that rely on stealth to find their prey or successfully hunt, may realize they've been seen and will give up a hunt. Or, these are not mutually exclusive, alarm calls could be directed to conspecifics to warn kin, to create pandemonium through which an individual could escape. Now, they're not mutually exclusive, and one of the really million-dollar questions I've been asking myself is, how can we tease apart the importance of each of these in the evolution of alarm communication and maybe in the current adaptive utility, how it's being used right now. Haven't gotten to the current adaptive utility, but I think we have cracked the evolution question. An honors student a number of years ago and I conducted a large literature survey looking at rodents. So we scored 209 species of rodents as being social or not, alarm calling or not, and being diurnal or not. And we used several phylogenetic and non-phylogenetic hypotheses or methods to seek to explain variation in whether a species alarm called or not. So in the phylogeny on the left, what you'll see is the 209 species of rodents we focused on, and some of these lines are black, and those black lines illustrate reconstructions of alarm calling being present, and the white lines illustrate reconstructions of alarm calling being absent, and the blue boxes, which you can maybe see a little bit, illustrate evolutionary gains of sociality, and the yellow boxes illustrate evolutionary gains of diurnality. So in these 209 species of marmots, the evolutionary origin of alarm calling was preceded by the evolutionary origin of diurnality. Individuals call when they're safe. It's not safe to call when you can't see and manage predation risk necessarily. This is consistent with the hypothesis that individuals are calling to signal detection. Diurnality doesn't precede sociality. Sociality explained less variation than diurnality in whether a species called, and species that evolved sociality were only marginally more likely to call. So while all of the cool stuff that we think about and things I've studied thinking about the current adaptive utility and the meaning of alarm calls to conspecifics, this is an exaptation. Calling in rodents seemingly originally evolved to talk to the predators. And I think the take-home message from this is calling is a game between predators and prey. And when you think about range expansions of predators, or you think about reintroductions of predators that might then, formerly naive prey may then suddenly interact with them, these novel predators may not be playing the same game as alarm calling species in those environments. And we have to think about this, and we could potentially study this to see if this makes a difference or explains some of the successes or failures that we have in reintroductions. Another game animals play is that all animals, eventually flee an approaching threat. But in addition to fleeing an approaching threat, if you're a refuging species, if you use a refuge as a burrow to find a safe place, or you hide in cover or something like that, you have to decide how long to remain in the burrow, how long to remain in your refuge. And people have thought about this as a game. If you spend too much time in your hole, you're going to starve to death. And if you spend too little time, the predator may still be there. So predators are playing a waiting game, and prey are playing a waiting game simultaneously. So let's explore this in yellow-bellied marmots. Well, it turns out that yellow-bellied marmot hiding time, how long they remain in their burrows after they've been scared into them, is sensitive to both the costs and benefits. So if we put food outside a burrow, food availability influences the decision to remain in their burrows. And the speed of approach, how threatening we were, also influences the decision to flee. Some models we've done suggest that there's substantial fitness consequences to these decisions. So marmots that weren't employing strategies that properly evaluated the costs and benefits of hiding would have lower fitness than those that would. So these results suggest that hiding time is sensitive to both benefits and costs, and it's likely to have co-evolved with predation risks from specific predators. Different predators might play different games with their prey. So animals experiencing different predators might not have that sophisticated set of rules that allow them to coexist, and we might be generally wary about the consequence of novel predators on prey, particularly when games like this are expected to happen. Sounds have to be transmitted through their environment and through space, during which time they can degrade or attenuate in a variety of ways. In particularly destructive environments, effective communication may have to be rather simple. Now, if that environmental structure or habitat acoustics makes it so that two sounds can't be discriminated between each other, and that's important for normal communication in a species, then it's sort of interfering with normal communication in a species. Janice Daniel and I spent 5 summers studying marmot habitat acoustics, where we recorded and digitized and rebroadcast alarm calls and pure tones through a lot of different species of marmots' habitats. And we looked at how these sounds changed. For alarm calls, we used a technique, and pure tones also, we used a technique called spectrogram correlation. What we're doing here is we're excuse me, playing an alarm call and then re-recording it at different distances, 10, 20, 30, 40 meters from the speaker, and then basically taking this call and sliding it over this call and seeing how much the overlap was. If the overlap was high, you would have a high spectrogram correlation. If the overlap is low, you would have a low spectrogram correlation. So here we see both processes, 2 main processes happening. We see attenuation, meaning the amplitude in this waveform is decreasing. Calls are getting softer as you go farther away. But importantly, we see degradation. The smear is very different from that initial thing. And the shape of this call is very different from the shape of the original call. So you can do this through different sorts of habitats. And you can also do this with pure tones. The dominant frequency of most marmot species alarm calls is about 3 kilohertz. So what we did was we broadcast 3 kilohertz calls through a bunch of different species habitats. And what we found was these pure tones had different correlations. The environment influenced them differently. There's substantial variation in a species acoustic environment. So some species, when broadcast 40 meters, 10 to 40 meters, the correlation value declined a lot, while other species it didn't decline much at all. So there's a lot of variation out there in habitat acoustics. Now, so because species live in specific acoustic environments, we should consider the acoustic environment when quantifying habitat quality, when selecting locations to release individuals, particularly for species that have biologically important vocalizations like singing or alarm calling or things like that. And again, set this up as an adaptive management experiment. See if it makes a difference. That's at least my suggestion. So variation in calling is profound. Not all individuals call when you're walking your dog around a particular habitat or through a particular habitat. Why? What's going on with this? From an ultimate perspective, it could be because there are different benefits or costs associated with calling in a particular situation or to a particular sort of predator. But what about from a proximate perspective? We used what I'll call the trap calling assay, where we noted when we trapped marmots, yellow-bellied marmots here, they call sometimes, and the individuals that call in traps are also more likely to call when we approach them or when they encounter a predator. So we used this trap calling assay, the frequency of calling in traps, and we sometimes marmots would call in a trap, and sometimes marmot wouldn't call in a trap, and sometimes, often, they defecate when they're in the trap. So we collected that, and we compared fecal glucocorticoid metabolites on 2 occasions, one when an adult female called and one on when an adult female didn't call. And in the final analysis, results weren't confounded by time of day, age, breeding status, or time of season, which are other factors which could affect calling. And those don't seem to, affect glucocorticoid levels, and those don't seem to in this case. And what we found was that glucocorticoids seem to potentiate alarm calling. We have not injected cort into animals to increase calling, but females that called on an occasion and didn't call on another occasion, on the occasion that they called, they had higher concentrations of fecal glucocorticoids. Their CORT, a stress hormone, was higher in their bodies, or that's what we infer from this. So what's this mean for conservation? Well, calling may be a risky behavior. I'm not sure it is, but we often assume that giving an alarm call is a risky behavior. And we assume that because, well, I mean, I find animals that alarm call. If I'm walking through a meadow and no one's calling, and I'm looking, I may not see an animal. The animal calls, I can find the animal. Predators, we assume, also find animals by alarm calling. So if calling is risky in that it exposes animals to predators, be very careful about stressing free-living animals out in whatever ways we might stress them out. It could potentiate calling. It could make them more likely to call and be really a mechanism or a pathway by which predation risk on those animals could increase. And second, calling can be an indicator of stress that can be used both in captivity and in the wild to monitor our management actions or to monitor the rearing conditions. And people have suggested this for, I think, squirrel monkeys, where squirrel monkey calls also seemingly are CORT potentiated, and that in captivity they were working with ways to better house squirrel monkeys, and by using alarm calls as sort of a non-invasive assay, they could try different techniques. We conducted an extensive set of habitat analyses on locations where yellow-bellied marmots were never seen over 40 years of study, where yellow-bellied marmots were seen over 40 years of study, and where yellow-bellied marmots went extinct at some point during those 40 years. This is one of the wonders of a long-term data set. You can actually begin to look at characteristics that might be associated with extinction. And we classified these two sorts of things into fear factors and food factors. So fear factors are the things that look at visibility or the steepness, which influences visibility, or the number of large rocks in a marmot's home, in an area where marmots are, which influence safety, particularly from badgers or animals that might dig marmots up. Grass cover and vegetation height were our two metrics of food availability. Admittedly, those are very rough metrics of food availability because marmots have a very Catholic set of food habits and eat a variety of different things. But we just use these as rough indices of habitat quality of habitat quality in this study. And when we've sort of fitted a logistic regression or a series of different models to try to explain whether populations persisted or went extinct, and or whether marmots were in a location or weren't in a location, what we found was that these fear factors explained significant variation in persistence or extinction, whereas the food factors explained no significant variation in persistence or extinction. Take-home message from this is that when describing the habitat and looking for places to reintroduce species, it's really essential to examine risk factors as well as food factors. Your introduction could fail if risk isn't properly considered. We need to introduce animals into places where they're going to be safe, and we need to think about safety from an individual's perspective. And by obsessing about food, which is an important thing because we know many reintroductions fail because animals starve, but we might neglect to think about fear and safety, and that might not be a good thing. Individuals vary in predictable ways, and people talk about personalities, which are suites of correlated behaviors. And you know, at one level, we've sort of recognized this. I mean, there's a rich literature on alternative strategies in the behavioral literature, at least. But at another level, we really haven't recognized these personality types as much as we should have until recently. And we've treated many differences between individuals as statistical noise. We use ANOVA to factor out individual differences. We try to look for other effects and ignore these individual differences. But anyone with a pet realizes that, you know, animals behave in consistent ways. And a number of people have begun to try to understand these personality types. I like personality. Some people think it's anthropomorphic. I think it's okay to be anthropomorphic in some ways, and in this way I think that's an okay thing. There are other ways of thinking about personalities or consistent phenotypic differences. Some people use that. Seems a little jargony for me, but it's okay. And there's another literature out there that begins looking at behavioral syndromes. Now when people are talking about behavioral syndromes, they're talking about correlations across different contexts about how animals behave. So it's not simply that animals, you know, have a suite of correlated behaviors. It's that in very different situations or in very different contexts, there's some linkage between these things. So, you know, people talk about in the context of anti-predator behavior, people have talked about shy, bold, or proactive, reactive syndromes. Now, in some cases, animals are systematically bold across many different contexts. They might be bold around males, might be bold around females. Males might also be bold around predators. And while you might think that it might be good to be bold around females, you might think it might be bad to be bold around predators. So understanding the nature of these syndromes and the structure of these syndromes might shed light on not only explaining natural variation, but might have some implications for captive rearing and reintroduction. Now, these syndromes themselves may be adaptive, or they might illustrate constraints on adaptive evolution. But again, regardless, to really understand anti-predator behavior, I suggest, and a number of other people suggest, that we really need to begin understanding and getting a handle on this individual variation and documenting personality types and looking at the fitness consequences of these and looking at the fitness consequences of syndromes. Now, personality does have fitness contrasts, and what I'd like to start by talking about is a meta-analysis that Brian Smith, one of my graduate students, and I conducted recently. Now, we know that bold foxes die following reintroductions, and we know that bold guppies get more sex. But generally, how important is boldness? Can we make a statement about that across a variety of different species? Not all studies where people have looked for fitness consequences of these sort of personality types have found them. And you might not expect them to, because at equilibrium, fitness should be the same. So if the equilibrium frequency of shy and bold individuals, or if the frequency of shy and bold individuals is at some equilibrium, the fitness of all of them should work out. So we conducted a meta-analysis. So meta-analyses are quantitative techniques that combine the results from previous studies to estimate a combined effect size. I do a lot of comparative analyses, and I've lost hair doing meta-analyses because whenever I'm doing a comparative analyses have all those sorts of issues associated with them. Meta-analyses are even worse because first, people don't report data. I mean, it's remarkable how little data are actually reported. People don't report sample sizes sometimes, or p-values are shoddily reported, and they just report ns. Or you can't find a standard deviation, but you can see a mean on a plot, or something like that. So to calculate to conduct a meta-analysis, you need to estimate effect sizes of individual studies. So that's one challenge. The other challenge is that really what you're doing is you're trying to somehow convince yourself, so it's always good to do this with someone else so you can convince yourself and they score them independently, and if you both agree, then you're okay. But somehow convince yourself that the same hypothesis is being tested. So if you're studying the effect of a blood pressure medication on blood pressure in different countries and for different sexes and different age people, that's a little easier, which is really what meta-analysis was initially created for. But if you're trying to study the fitness consequences of personality, and people are studying these things in very, very, very different ways, can we really say this is testing the same hypothesis? So Brian went through a stack of about 1,200 papers, and we ended up with 12 studies. But we had other help with this too. But we asked two key questions. Do bolder animals have higher reproductive success and lower survival? So there are a number of problems with this because people have sort of conflated the use of personality and behavioral syndromes. And mostly here we're looking at behavioral types, which might be a personality rather than a behavioral syndrome. And as I said, people don't report their data, which is kind of annoying. What we found was that bolder mammals have greater reproductive success. And we also found, and so this is small, but it's a positive effect. But we found no effect of boldness on survival. So bolder animals don't live less, or they're not more likely to die than shy animals who are more likely to breed. So really, populations can consist of both shy and bold animals. And maybe we need to maintain this behavioral variability when we recreate populations and reintroduce populations. And if we don't ask the question and try to document this stuff before we recover a population, we're never going to keep and capture this variability. I'm an empiricist. I mean, the initial comments, paper was initially rejected because they said N of 12. Yeah, we looked at like 1,200 studies, and these were the studies we could confidently extract data from. A number of other people are doing this work now. We actually looked at birds also and other species, but for the mammal, the sample size was 12. And And other people are doing this, and maybe these results will change, but that's what science is. So in any comparative study or any meta-analysis, hopefully it can stimulate research, and hopefully it will stimulate people to report data that can be used in future meta-analyses. So that's the story now. It may change in the future, but that's okay. I now start all of my classes when I'm teaching by saying, you know, most of what I'm going to tell you is wrong, and I don't know what it is, but hopefully in 20 years we'll all realize what it is, because that's how we learn. In addition to studying predator recognition abilities in captivity, which I've done with Vancouver Island marmots, because they were one of the main hypotheses for their decline is predation by not novel predators, but predation by predators. We also were looking at personality types, and we were doing this by, and also syndromes, by using a technique called mirror image stimulation. Put a mirror in a cage, see how animals respond. And we did this with a stuffed taxidermic wolf present or no wolf present. And we began scoring behavior 30 seconds prior to the marmot discovering this mirror, and we scored behavior for the next 10 minutes. And we looked at foraging, vigilance, interactions with the mirror. I'll show you some videos in a second. Tail flagging, and escape into their burrows. And we used principal component analysis to extract orthogonal factors. So here's a Vancouver Island marmot. Look at the red spot. And it's investigating a mirror. At least this one is. So it's rubbing up against the mirror. It's touching it with its nose. It's going to tail flag a little bit. This is a strong investigator. Who is that in my cage? Not all of them are. Follow this guy. This is its first exposure to the mirror. We put the mirror by the food bowl, looks at itself, really looks at itself, greets itself. Very different response. And this continues. Tail flags a little. So, and you can sort animals out. So what I'm showing you is, on the left, there's the mirror image stimulation, MIS, without the wolf present, and on the right, the MIS with the wolf present. So these are rotated factor scores. And this illustrates several things. The bold values are important in interpreting the factor scores. So you'll notice that the factors are somewhat different when we have a wolf present and when we have a wolf not present. However, having said that, let's just look at factor 1 for a second. If we look at factor 1 from the mirror image stimulation experiment, we see that the first factor probably can be interpreted as an interactive factor. There's not a lot of vigilance. It interacts with the mirror. There's a lot of total foraging. The MIS wolf factor, it interacts with the mirror. It moves around a bit. It tail flags a lot. Tail flagging is a social sort of behavior, and it's not in its burrow much. While the loadings are different, we probably can interpret those factor 1 as being somewhat of an interactive factor. Now these factors explain 72% of the variation in responses, which is kind of nice. And that while the set of marmots that had both of these, MIS and MIS wolves, was small, 14 individuals, we had a larger set of responses with the MIS alone. When we looked at those that were studied the same, we more or less had identical results. And if we assume these results are somewhat robust, we also see that about 25% of the variation in how a marmot responds to a mirror plus the wolf can be predicted as a function of how it responds to the mirror alone. So if we're trying to come up with ways to sort of potentially survey animals in an easy way before we release animals, maybe we can get something from this mirror. And there are correlations with how animals respond to a mirror and to a mirror with a wolf. So because there's a correlation here, R is 0.5, we've identified a syndrome. What is it? Well, maybe it's a mirror syndrome. Now that itself seems kind of artificial, but it does illustrate that by showing a mirror, we can have some predictive value. What about how this tells us about how animals respond to predators? Can the mirror without a wolf response tell us something about predator recognition? So as I said, we presented a variety of stimuli to marmots in a predator discrimination experiment in captivity. And what we found is that So what this is illustrating is the correlation coefficients of factor 1 score in the MIS experiment and the responses of the marmots to different predators or control stimuli. So remember, factor 1 is sort of this interactive factor. Interactiveness is correlated with how much time the subject was vigilant at or in the burrow when a cougar was present. It was also positively related to this response when the cart and the marmot were present. So when a cart, all these predators were sort of rolled down on a cart and suddenly came into view for the marmots, and one of our controls was a marmot. So when there's a cart, it's kind of a novel kind of thing, the marmots explored that. And when a marmot was present, they explored the stuffed marmot. But they also responded aversively to the cougar, and how marmots responded to this mirror was somewhat predictive of how they would respond to a mirror. So how's this at all relevant for conservation? Well, at the last time I looked, 4 of these pre-screened animals were released. 2 of them were killed, and the male that was the most interactive was killed by a cougar. So maybe if we can bootstrap up this data set, and we can talk later about why it's sort of, I'm not bootstrapping up this data set and why it's hard to do this sort of work, we could have predictive value, and we could try to know something about variation in behavioral types before we introduce animals, and we have ways of potentially, in an adaptive management context, develop a data set that will be useful for picking which animals might survive and which animals which may not. So what if we find certain animals aren't going to survive when being released? Well, what do? Well, we could selectively release animals. We could potentially pre-release train animals to enhance their responsiveness to predators. We could remove predators, which in some contexts is okay and in other contexts isn't okay at all. Or we could remove other reasons that predators are there. In the case of the Vancouver Island marmots, one of the causes, or one of the hypothesized causes of the decline is a hyperpredation hypothesis, whereby high altitude logging brought in a bunch of deer. The wolves and cougars followed the deer. The trees are growing back. The habitat's becoming less suitable for the deer. Eventually, the wolves and cougars will leave, and this will be safer for marmots. Now, with yellow-bellied marmots, I wanted to address a more general question of wariness and locomotor ability. Specifically, are slow marmots likely to be generally wary? Now, there's several ways to think about this question. Some species, when they're in exposed places, reduce their overall exposure by spending less time there. So, I live in LA, where you don't really want to go to some ATMs at times, and there are different strategies one could adopt when going to a dangerous ATM. You could spend a lot of time in your car looking around, look, and then run in and run out. That's one time minimizing strategy. When you get to the ATM, you could be looking around a lot. That's a awareness increasing strategy, but it's sort of increasing your exposure because you're at the ATM longer. Or you could get a bunch of friends and go in as a group and run at the slightest provocation. And animals have all of these and other strategies to deal with foraging or doing things in risky places. So if we could find a relationship between how marmots foraged and how sort of locomotorily challenged they were, we could begin to define a syndrome. And we looked at wariness several ways. We looked at how close a marmot would tolerate an approach by a human, and we looked at how much time marmots allocated to anti-predator vigilance while foraging. And what we found was that slow marmots minimized exposure by foraging more and looking less. Now, what this illustrates is residuals, and it illustrates residuals from 2 GLMs. In order to study how fast marmots run, we actually have a model that predicts run time. And that's because we are interested in looking at distance run. So velocity is distance over time. And there's something called the ratio correlation problem, which is a statistical problem where you can't have something, you can't regress distance against distance over time. You're going to get a spurious positive correlation. So the way to crack that one is to sort of say, well, let's model running time, and let's model running time as the amount of distance you run, and animals' age, and sex, and body mass, and substrate, and incline, and a bunch of those things. And then after throwing all these things in, we can predict running time, and we can look at an individual's running time, and we have our prediction equation, and we can say the residual variation is that intrinsic locomotor ability that marmots may have. So there are fast marmots and slow marmots. And similarly, for time allocation for foraging or vigilance, there are a bunch of things in the environment that influence these. percent cover, group size, incline, other sorts of habitat factors. So we created a general linear model there, and again, sort of looked at the intrinsic foraging veracity or intrinsic vigilance of different individuals. And again, we found that slow marmots minimize exposure by foraging more and looking less. So the fact that these are related suggests that there is a locomotor ability wariness syndrome. At this point, I don't know what the fitness consequences of this might be, but I'm trying to study that. And I wanted to sort of take this vigilance thing and go with this. So the next question was, how is vigilance while foraging related to response to novelty and to behavioral responses in a trap? So we conducted some additional experiments where we looked at the responses of marmots to a variety of different experimental treatments. One of those was we put food in a goose, we put food in a mirror in the field to try to collect personality or syndrome type data that we could use. And then when we trap marmots, sometimes they fight in the trap and call, sometimes they defecate, sometimes they struggle, sometimes they just sort of sit there and wait to be processed. So we collect this information routinely when we trap them. And I suggest that if you're trapping animals, please record everything you can contextually about that trapping thing. You can get some interesting, repeatable, and potentially predictive data from those sorts of experiences. So I'm not going to show you data. I'm going to show the preliminary synthesis, and it doesn't make a lot of sense at the moment, but we're working on this. So, I've demonstrated a maximum running speed wariness syndrome. Slow marmots are less vigilant while foraging. They're dining and dashers. We then found a relationship between vigilance while foraging and vigilance with a goose. Now, this goose seemingly was the crux for all of this, because how vigilant animals were with a goose also explained how vigilant they were when you just put food there. Maybe they didn't see the goose. I don't know. I mean, this goose is right next to the food. And it was negatively correlated with how relaxed they were in a trap. So at least provisionally, I'm suggesting there might be some sort of a novelty syndrome here. Let's come back to sort of conservation behavior and say, okay, well, this is fine and good. What are the take-home messages? We should expect syndromes. We should look for syndromes. And I believe we should design adaptive management reintroductions where we can quantify traits that are going to allow us to look at the consequences of these things and see if it makes a difference. If it does, then maybe we can increase our reintroduction success. If it doesn't, it was a nice experiment, and go on and do other things to try to increase reintroduction success. Living socially is not all what it's cracked up to be. Animals may be exposed to more parasites and pathogens. It may be stressful to be social. Think about, going home for holidays, after you've gone to college or something the first time, oh, do I really want to be here? Maybe you did, maybe you didn't. Animals are competing for food resources. There might be reproductive competition. All this stuff happens when animals are social. Now, let's think about reproductive competition. Life history traits reflect investment decision animals make about allocating energy to growth or reproduction. From the literature, Ken Armitage and I, a number of years ago, looked at siluriid rodents, marmots, prairie dogs, and ground squirrels, and we extracted a bunch of life history traits: age at first reproduction, percent survival to age 1, the percent adult females that are breeding, and litter size, and we created a metric of social complexity. I'm not going to talk about this metric here, but I'm going to show you some results. These are the results of an independent contrast analysis where more social, so what this shows is that the evolution of sociality is associated with the evolution of reproductive suppression. More social animals have fewer breeding individuals in the group. Socially complex species delay the onset of reproduction. The age at first reproduction is delayed. And that socially complex species have fewer young, but they seemingly invest more in them. So the relationships between sociality and demographic things that we care about, age at first reproduction, survival, percent adult females that breed. Okay. So while a lot of people think about causes of social complexity or causes of sociality, sociality itself has consequences that we as conservation biologists and managers should care about, because many of these are demographic consequences. And this isn't expected. Sociality has demographic consequences. And these are some of them, at least in siluriid rodents. Well, Ken initially has been looking at a lot of characteristics associated with breeding in yellow-bellied marmots. and has found that individuals don't breed if they have insufficient body mass. That's fine. They're climatic correlates of breeding. But importantly, they're also 2- and 3-year-old marmots may not breed. They may be reproductively suppressed. Once an individual reaches 4, she's going to breed at whatever frequency she breeds. So why would an individual tolerate suppression? So there's animals which have what's called age-specific reproductive value, the average amount of future reproductive success for an individual of a given age and sex. Now we know that it varies by age and sex. Just think of female humans. If they've got a ticking biological clock, that's because they're going to soon not be able to reproduce anymore. And we know that men don't typically have this problem. Men can breed, particularly now with Viagra, for an extended lifespan. Now, there are a couple of components associated with this. There's total reproductive value equals present reproductive value, what's allocated now, and what remains following current reproduction. This is called residual reproductive value. George Williams suggested that for any reproductive attempt, selection should favor individuals who maximize their current probability of reproductive success with a minimal amount of effort and risk. By doing so, Williams suggested that they're going to maximize the residual reproductive value as well. So individuals should suppress themselves if by reproducing now they actually harm their residual reproductive value while having a low probability of breeding success. So reproductive suppression should be favored when the reproductive success at a future time minus the reproductive success now is greater than whatever that cost of suppression is times the residual reproductive value. So we would predict individuals potentially, if this inequality is true, to suppress themselves. It's all about future reproductive success. And we may see individuals, females, reproducing. While we may see females reproducing when it looks pretty bad out there now, maybe it's also pretty bad out there in the future. But if it's bad now, but if the future is brighter, it may make sense ultimately to not reproduce now, particularly if reproducing now is costly. Okay, so take-home lessons initially are complex sociality is characterized by reproductive suppression. And there are a number of mechanisms that Wasser and Barish suggest that might be responsible for this. Animals could delay sexual maturation. This is seen in a variety of rodents and some primates. And the sort of important sociobiological question is how much of this is influenced by variation in food resources and how much of this is socially induced. In some cases it's socially induced, sometimes by the mother, other times by other breeding individuals. And delayed maturation may be also an effective mechanism to prevent inbreeding. So this doesn't seem to be happening in yellow-bellied marmots. There could be inhibition or delay of sexual receptivity. We're looking at this. Subordinate wolves, mice, and wild dogs, for example, may be sexually mature but not sexually receptive if they're not the dominant individual. I don't think this is likely in yellow-bellieds because we see evidence of females cycling. We see nipples developing. We see other correlates. We think of external correlates of pregnancy, yet we don't see any young emerge from the burrow. There could be inhibition or delay of ovulation. Rodents and a variety of primates may fail to ovulate if they're subordinate. Again, this doesn't seem to be happening in yellow-bellied marmots because we see marmots who are lactating or with very developed nipples, but we never see a litter emerge above ground. There could be failure of implantation, and we're trying to crack this one physiologically. If we can catch animals enough, we can follow hormonal pregnancy profiles in blood or feces, and we can ultimately shed some light into this. We don't know right now, but we know that spontaneous abortion is well reported in a variety of rodents, a variety of marmots, and possibly in yellow-bellied marmots. And again, if we can get enough sensitivity on following hormonal pregnancy profiles, we might be able to nail this one. There also could be early postpartum mortality. This might be possible. And individuals of many species may tactically not care for their young if by doing so they're harming their future reproductive success. There's something called the Concorde fallacy where when the British and French were building Concorde in the 70s, at some point they realized they never would make enough money by doing this, but they continued doing it anyway because their national pride was sucked up in this. Animals are predicted not to commit the Concorde fallacy, so there might be an early postpartum mortality. And finally, infanticide is not uncommon in marmots. Infanticide by females is seen in a variety of rodents, including yellow-bellied marmots. We don't think it's that common. Could be. Really hard to sort of detect, but we're looking for evidence of this. So I'm talking a lot about a lot of mechanisms, mechanisms by which animals may suppress themselves. Why is this important? Well, at one level, it can illustrate whether suppression is something that's forced upon individuals or something that they do to reduce costs. This is theoretically interesting, but it also can illustrate wasted reproductive effort. And minimizing waste and not burning up an individual's residual reproductive value is particularly important in captive breeding situations if we want them to, be fruitful and multiply for a while. So I believe that understanding these mechanisms for any situation where you've got captive breeding where this might be going on is going to be important. If infanticide is a mechanism, there are a number of strategies that both in captive and wild populations that you can do to mitigate this or prevent this. So I've talked about infanticide by females, but there's also sexually selected infanticide by males, which people study in bears and things like that. Lions. Known mechanisms can be managed. Particularly if infanticide is an important mechanism, then there are things you can do to prevent it. You can keep breeders apart. You don't provide the opportunity for infanticide. And again, knowledge is power in this case, and documenting these things and understanding these mechanisms may be pretty important. So a lot of people helped out with this over a number of years and a number of different places, and in the past at least, a variety of funding sources. Now I'm having some issues getting funding. Which is ironic because I think there are a lot of benefits associated with supporting long-term research on marked individuals. We can identify factors that drive population processes. We know that juvenile survival is very important in the marmots, for example. We can study rare phenomena. Reproductive suppression doesn't happen every year. Doesn't seem to happen in good years. It doesn't happen when I've spent a lot of time and money trying to study it. Everyone's been breeding. Our population's been increasing. But it can happen at times, and during those times it can be pretty important. You can study climatic effects on behavior. The marmots at the Rocky Mountain Biological Laboratory are emerging earlier, and in some years through more snow. And there's higher spring mortality in those years where they're emerging earlier through more snow. We can identify links between different aspects of behavior. We can really begin pulling together what syndromes exist. And we can study senescence and evolution. Is it important in natural selection, in natural populations? What traits are under selection? And I'm beginning to study some of those questions. And as I said before, we can identify correlates of extinction, which really requires long-term data sets. So to remind you and summarize and wrap up, I think some lessons from marmots that are relevant for other questions, potentially, are anti-predator behavior evolved as a game between predators and prey. Different predators may not play the same game. There's substantial variability in a species' acoustic environment. This must be acknowledged when selecting reintroduction sites, particularly for animals that need their communication. Glucocorticoids may potentiate alarm calling. Calling is risky. Be careful about stressing animals out, at least to increase their exposure, or use that as a metric of exposure or stress. Predation risk may influence population persistence. If so, it's essential to quantify risk factors as well as food factors when identifying release sites. Individuality may have fitness consequences. Please try to quantify this stuff before you reintroduce animals. And complex sociality is characterized by reproductive suppression. Anticipate suppression. Try to identify its mechanisms and try to mitigate these mechanisms when you can. So I guess I still was able to talk, maybe too much. Thanks for listening, and I'd be delighted to try to answer some questions.
Dave, I was wondering, since you said you had, it's like a long-term data set with individually marked animals, I was wondering if you'd sort of follow up on the, looking at the alarm calling to see if indeed the marked individuals who you know are callers, if they have sort of lifetimes less survival.
We're looking into that. Looking into that soon. That's one of the things I want to get to soon. Another interesting question that you can get at with that is that recently Chris Evans' group in Macquarie University in Sydney has demonstrated that chickens, male cockerels that give high-risk alarm calls, females like it. Alarm calling is maybe under sexual selection, which no one's ever thought about before. So I also want to, with our reproductive success data, which we're trying to work out now, I want to begin looking at that and other questions associated with longer-term consequences of calling more or less. Yeah?
You talked about using behavioral syndromes to predict which individuals would be best in reproduction. I'm wondering, first, how often do you
We do it all the time with genetics, don't we?
I don't know.
Well, we try to.
Have surplus?
We try to create mixes of individuals that are going to maximize effective population size.
And release.
And release not close relatives and try to have outbred groups, et cetera.
Yeah. So that relates really closely to my second question, which is, or I guess maybe if you're releasing all of them anyways, it doesn't matter. But I was just thinking that if you are behaviors related to genetics, it's terrible. These syndromes are terrible. And if you are selecting in your captive breeding program or in your reproductions for certain syndrome types, are you going to be reducing genetic variability?
That's an outstanding question, and you nailed the sort of what I think is the sort of the key bugaboo with this knowledge. You know, we play God all the time. And let's just say you find that a personality type or a particular syndrome is there, and that individuals on one end of that syndrome, you know, might be more vulnerable to predators and predation. Do you not release them? Then you don't maintain that variability if this is heritable variation. I don't know. I mean, I think we're beginning to have those conversations, and it's not obvious. I think correlates of translocation success are having lots of warm bodies or cold slithering things to reintroduce at one time and over time. So anytime when you don't have a big pool, big reservoir of individuals, your reintroduction is not likely to succeed. You really got to throw a lot at it. And maybe it's a lot at one time, maybe it's a lot over time, but these are ongoing processes. These use a lot of money typically, and most people getting into captive breeding and reintroduction realize that they're going to be spending a lot of resources. So at that level, I think it makes sense to begin to at least collect these data. And you know, I don't know. I mean, if we want to maximize the variation, maybe we want to maximize the variation of survivors. So it's a really tough question. I think you nailed it. I don't have the answer to that. Yeah.
These are truly personality traits. I assume that they'd be consistent in an individual across multiple recaptures. Have you mapped that to see if Cooper's always a Cooper?
We've done some analyses of the repeatability of these, and we want to do more. In other systems, people have seen these sorts of things, and so there's studies of great tits in Europe where people have done this, and there it's easy. You just go to a nest box and take an animal out and put it in a scenario and look at it over time. Ken's done mirror image stimulation tests sort of in the field where he's taken animals out and put them in a little captive enclosure there, and he's found that individuals were repeatable within an age class but not across age classes. I'm using different techniques to try to sort of look at the ontogeny of those things as well. So that's a very good question. So the way I'm looking at this trap stuff ultimately is by thinking about repeatability.
Do you think that the researcher intervention is having an influence on how they respond to being trapped, how they respond to being trapped, or the normal where they're being?
Well, these animals have been trapped since day one. Yeah, I mean, you know, so it's sort of the Eisenberg uncertainty principle. You can't study a system without sort of influencing it, right? So I don't know how to answer that properly. I will say there's a lot of variation in how these animals respond when trapped. Even those animals, I mean, we have some animals that always go into traps and alarm call every time we come up to the trap. What's that about? We have animals that go into traps all the time and sit there like a log. We have animals that clearly learn how to be easy and clearly learn how to be hard, which isn't very nice. And then we have this really bad pernicious behavior where mothers are teaching their kids how to roll traps and get the bait from under it. And you know, that's leading to the unstudied marmots. So that's a really nasty thing. And we've seen this start in one group. And basically the daughters sort of who end up sticking around learn this. And thank God they haven't dispersed to other groups yet. So it's really a pain in the butt to capture animals in one of these groups because of this horrible behavior. But I don't know. I mean, it's sort of one of those unanswerable questions. I mean, we trap a lot to keep the mark, to get samples, to try to catch new animals that are moving in, and we have very spotty trap records. Some animals, we catch pretty much everybody at least once a year. We catch some animals 20 times a year. We catch a lot of animals about 4 times a year. Yeah?
Do individuals that have high court responses always have high court responses?
I don't know. No, because we looked at individuals, the same individual on 2 different occasions and controlled for a whole bunch of other stuff, and basically said, you know, what's the effect of what's the relationship between whether it called in a trap or didn't call in a trap, and it was higher court when it called in a trap. So it's a match pair study. So it's not that they're always high, and there's a lot of variation in court, and I'm trying to understand what's going on with that.
The relation between court and DeMark is always that, well, stress might be bad, but at the same time, that's a response of dealing with a stressor.
These were all breeding females. Now, so
how does it affect survival?
Excuse me?
If they're all breeders, then does higher alarm calling have a negative effect on survival or a positive effect on survival?
well, it may have a higher effect on fitness. So, you know, females that wean young are uniquely more likely, when a young emerge above ground, so they're born in the burrow, they're pregnant for about 34 days, they give birth in the burrow, the young are in the burrow for about 34 days, and they emerge out of the burrow. And when they emerge, they're more or less weaned. Uniquely, females that have young that have just emerged are much more likely to call. So all of these were adult females. I remember all of them were adult females with kids. I can't remember. Or all of them were pregnant or something like that. So I was hoping that court would be a mechanism of suppression, but in at least our first analysis of this, we didn't have a lot of suppressed females. Almost every 2- and 3-year-old was breeding. And in our first analysis of this, it wasn't really consistent with that. But I've got more data that I am just beginning to look at now and hope to understand that a little better. Interestingly, you know, there are things that should be influencing court that don't seem to be. Seasonality, we don't have strong seasonal effects on court. And again, I've got to look at this with this new data set as well. But food type can influence court. Time since last eating. You know, we have lots of variation. Not all of it was significant where we sort of expected it to be. And we're also doing a whole set of studies looking at the relationship between court and immunosuppression, which we're looking at by looking at neutrophil to lymphocyte ratios, white blood cell types, and also looking at a variety of intestinal and ectoparasites these animals have. So we get parasite diversity measures, and we're trying to sort of ultimately put together whether and how there are costs of sociality. I thought I had an answer. I don't think that's robust, and we're just throwing more data at it. So my initial working hypothesis was they don't want to be social, and they're badgers, and European badgers. European badgers live together, but they don't really want to live together. I think that's yellow-bellied marmots, but I'm not sure. Some lines of evidence suggest that it is, some lines don't. But that's the assumption I'm working under.